Thank you, Dan, for that generous introduction. I'm deeply honored and delighted to receive the ARO Award of Merit. I'd like to thank the Society for this distinction and hope that I can reach the high bar set for this lecture by previous winners.
It listens for the tiny sounds of its prey moving about below, but must take great care not to scare them away.
This short video illustrates one of the premier examples of sound localization ability. I wish that I could say that I will be showing you behavioral data using this animal model. In Wisconsin, we have foxes and plenty of snow and rodents, but the behavior I'll show you is much less exciting. So here's an outline of the talk. I will begin with a brief review of the pertinent properties of auditory nerve fibers, and then review the cell types in the cochlear nucleus and superior olive that are thought to be important for this task. Then I'll spend most of my time on the physiological and behavioral studies from my lab aimed at understanding this intriguing ability. Since all of the information needed for neural computation of acoustic space must be contained in the auditory nerve, it makes sense to start there. So auditory nerve fibers have two important properties illustrated in the slide. They are sharply tuned in frequency with different CFs. Here we show frequency tuning curves of many auditory nerve fibers. CFs depend on the location of the inner hair cell that the auditory nerve fiber innervates along the base to apical extent of the cochlea. And in B, the responses to a pure tone burst at CF, auditory nerve fibers show a stereotypical response with either rapid or slow adaptation. For low, but not for high frequency auditory nerve fibers, there is phase locking, which is a response to each cycle of the sinusoidal tone, as shown below. This is seen more clearly in the next slide. So not surprisingly, the phase lock response of auditory nerve fibers results from the cyclic response of the inner hair cell, which is clearly present at low frequencies. Here we show low frequencies up to 1K and then 2, 3, and higher frequencies, where it disappears. The cyclic response disappears at higher frequencies, but there's still a large DC component. So the cell at high frequencies, the inner hair cell still responding, as does the auditory nerve, but it loses the cyclic AC component, which defines phase locking. Single unit recordings from auditory nerve fibers are plotted on the right in the form of period histograms. Here, the abscissa is one cycle of the sinusoidal input, here plotted in microseconds. The histograms show the preference for discharging over a range of phase angles, which gradually becomes less pronounced as we move to higher frequencies. So here we show 300, 400, 900, 1,000, up to 1,400 hertz. In the cat, phase locking essentially disappears above about 3 or 4 kilohertz. So here is Ramonica Hall's drawing of the projections of the auditory nerve fibers into the cochlear nucleus, showing how all auditory nerve fibers bifurcate and send one process into the DCN, BVCN, and one process into the ABCN. In the ABCN, there are some elaborate end bulbs of Held, which synapse onto the bushy cells in the BCN, which are the important topic for today. So you can see that cells in the BCN and DCN differ in the strength of the input from auditory nerve fibers. Furthermore, this schematic diagram shows that there's further difference in the elaborate circuitry in the DCN as compared with the VCN. We will actually only be concerned with the two flavors of bushy cells, spherical and globular bushy cells shown here, which receive that strong end bulb withheld input from the auditory nerve. So along with differences in the intrinsic properties of the cells in the DCN versus VCN, these differences produce very different response patterns, as shown in the next slide. Here we show how the post-stimulus time histogram and frequency tuning curves of high and low frequency cells, here high frequency, here low frequency cells that are phase locking. And this is typical responses to the auditory nerve and how they're transformed by the different cell types in the cochlear nucleus. So really, as I said, we're really going to only concern ourselves with the spherical and globular bushy cells. But it's interesting to note how the other cells in the cochlear nucleus are transformed in very dramatic ways, both in terms of their frequency tuning curves and in their PST histograms. As we'll see, there are also some important differences in the low frequency bushy cells, those plotted here, versus their responses in the auditory nerve. This table summarizes the properties of cochlear nucleus cell types and their projections into the auditory brainstem by the trapezoid body or the various other acoustic strias. So note that the spherical and globular bushy cells exit by way of the trapezoid body and project to the major nuclei in the superior olive, LSO, MSO, and MNTB. So let's digress for a moment and consider how stimuli are localized in different sensory systems. In the visual and somatosensory systems, space is encoded on the sensory epithelium, on the retina for vision, and on the body for somatosensation. The contralateral representation of space then is simply a matter of having the correct fibers cross the midline. But in the auditory system, the cochlea maps frequency, not space. So auditory localization requires a neural computation using acoustic cues. In the somatosensory system, touch input from the left hand reaches the right somatosensory thalamus and cortex, and by simply crossing at the midline, interestingly, the different modalities cross at different places. So light touch crosses in the medulla, pain and temperature fibers cross at their entry point in the spinal cord. In the visual system, an example of the contralateral representation can be seen as results of lesions of the right primary cortex, diagrammed here, which causes a loss of vision in the left visual field of both eyes, as shown here. This is set up at the optic chiasm by the nasal retinal ganglion cells crossing, while the retinal ganglion cells on the temporal side do not cross. So on the motor side, cells in the right motor cortex control the muscles on the left side of the body by a decussation in the pyramids in the lower medulla. So note in all of these systems, the contralateral bias is established anatomically by projecting the axon to the correct side. But auditory space is set up by a neural computation, not by anatomical projections. How is this done? To answer that, we have to look in the superior olivary complex. Here's Cahal's famous rendition of the superior olivary complex from Golgi preparations in the cat. You can see the three major nuclei, the lateral superior olive, medial superior olive, and the medial nucleus of the trapezoid body. Cahal also included the calyx upheld, an important synapse onto cells in the MNTB that will figure prominently later in this talk. So there are two circuits in the superior olivary complex, in the MSO and in the LSO, that are traditionally thought to encode ITDs or ILDs, respectively. Both originate from bushy cells. So cells in the MSO receive excitatory input from the spherical bushy cells on both sides, thus forming what we'll call an E interaction, excitatory. Cells in the LSO receive excitatory input from the spherical bushy cells on the ipsilateral side, but they receive inhibitory input relayed through an inhibitory interneuron in the MNTB that begins from the globular bushy cells projection to the MNTB. So we refer to this circuit as an IE interaction. And we'll see that it may be more appropriate to refer to these circuits as processing EE and IE rather than ITD or ILD. For the MSO, anatomical considerations would predict that the contralateral input will arrive later at the MSO than the ipsilateral, since it has a longer path length. Thus, you would expect the coincident cells in the MSO to respond best when the contralateral ear is leading, that is, if a sound source is on the contralateral side, to compensate for the longer conduction time. And this is what is almost universally found. In the free field, these cells in the MSO would be responding to sounds in the contralateral sound field. And MSO cells then project their axons to the ipsilateral inferior colliculus, thereby setting up a contralateral bias at that level. On the other hand, cells in the IE cells in the LSO are excited by sounds in the ipsi ear and inhibited by sounds in the contra ear. Therefore, they would be expected to respond preferentially to stimuli in the ipsi sound field. The nervous system converts that ipsi bias by sending those glutamatergic, presumably excitatory axons to the contralateral IC. Now in the cat, there are also a cohort of LSO cells that project to the ipsilateral IC. But some of these are glycinergic and inhibitory, as shown here, which would reinforce the contra bias in the ICC, while others, not drawn here, are glutamatergic. The result of all this is that cells in the ICC and higher centers would be predominantly, but not wholly, responding to sounds in the contra sound field. Note that in both the AVCN and in the superior olivary complex, some cells project ipsilaterally, some cells project contralaterally. Here for the AVCN, in the olive, in superior olive, there are ipsilateral projections and there are contralateral projections. So the anatomy itself is not enough to deduce the bias. One needs to know the physiology of the cells. Glendinning and Masterton and their colleagues referred to this complicated circuitry as the acoustic chiasm. Now, an additional complication is the MSO also receives inhibitory input from the MNTB and the LNTB, relaying inhibition from the contralateral and ipsilateral side, respectively. Now, the function of these inhibitory inputs to the MSO is highly controversial. And in the interest of national and international unity after 4 years of long national nightmare, I will defer discussion of this inhibition since I don't really have anything to add to this interesting question. So here's a nisosane section from one of our experimental animals in which an electrode penetration was made from the ventral brainstem through the different nuclei of the SOC. So in passing through the superior olive, one encounters many of the classic cell types. So for example, first you pass through trapezoid body fibers, which are monaural and driven by either ear, more or less randomly, and cover all CFs. Then you get to the MNTB, which cells are also monaural, but now driven only by the contralateral ear and at high CFs. In addition, these cells in the MNTB show a prepotential, a reflection of that large calyceal ending that they receive. Then, if you're lucky, you record cells in the MSO, which are binaural and have EE interaction, so therefore sensitive to ITDs, and predominantly low frequency. And finally, you reach the LSO, where you can see a lesion that we made in one of our experimental animals, where we recorded from a cell in the LSO. These cells are now IE, inhibited by contralateral ear, and sensitive to ILDs. And these cells in the LSO are predominantly high frequency, but if you get into this lateral limb, you will hit low frequency cells. So now let's consider the acoustic cues to sound localization. Along the horizontal dimension, space is computed using the two interaural cues of interaural time difference and interaural level difference. A sound to my right will reach my right ear slightly before it reaches my left ear, thereby creating an ITD, which can help encode its direction. Remarkably, the just noticeable difference for humans of ITD is 10 microseconds, extraordinary resolution. The sound to my right will also be louder to my right ear than to my left, provided that the wavelengths are short with respect to the dimensions of the head, that is, if they're high frequency sounds, so that the head can act as an effective acoustic shadow. So ILDs are predominantly a high frequency cue, and these constitute the so-called classical duplex theory of sound localization. Now, these acoustic cues can be quantified by measurements of the HRTFs, or head-related transfer functions, shown here for the cat for 5 points in space in the horizontal plane. So a microphone is placed near the tympanic membrane of each ear while playing a click from, in this case, 5 different points. The left and right ear signals are shown in blue and red. So for the point directly opposite the left ear signal arrives earlier than the right ear signal as expected, thereby creating an ITD. You can see that in the cat, this ITD is approximately 400 or 500 microseconds. It's also louder to the left ear than the right. It's reflected in the spectra, which are shown here. So the frequency spectra is higher, the gain is higher for the left ear than the right, and this is essentially the ILD function. Notice the ILD function is quite a complex function of frequency. A converse applies for sounds opposite the right ear, where the right ear signal arrives earlier and is louder. For sounds that are directly on the midline, the left and right ear signals are essentially identical, assuming a symmetrical head. So there are no ITDs or ILDs, that is, ILD, both variables are 0. So this is true for all points on the mid-sagittal plane. So even though we can discriminate whether it's the neighbor living in the apartment above us or below us that has the radio on, that is, we can localize vertically on the mid-sagittal plane. So that suggests there must be another cue that allows that discrimination when ITDs and ILDs are both 0. And there's now substantial evidence that the details of the spectra are a critical cue for vertical localization. And in particular, interest has been focused on this so-called first notch, which systematically moves in frequency as the sound source moves lower on the vertical plane. So I won't address the neural coding of these pinnacues, but we will touch on them briefly in a behavioral study later in the talk. So here's an example of how these HRTFs can be used to accurately simulate sounds in free field, so-called virtual sounds, which even produces percept that the sounds are outside the head. So this is a study of spatial receptive fields of cells in the LSO. So in the normal condition, the appropriate left and right ear HRTFs are used to filter the broadband noise and then delivered for 21 different azimuthal positions, varying on the horizontal plane from -90 degrees in the ipsilateral field to +90 in the contour field. The dot rasters show plot 20 representation at each azimuth, and as expected, the LSO cell response, so the sound is on from 0 to 200 milliseconds, right, turned off at 200. And you can see there's a robust response when the sound is in, simulated sound is in the ipsilateral sound field and inhibition for sounds in the contralateral field. This is plotted below in the binaural normal response. So this confirms the statement I made earlier that cells in the LSO are responding to sounds in the IPSI sound field. So one of the advantages of using virtual stimuli is that we can easily demonstrate the effects of the contralateral inhibition by turning it off so that only the ipsilateral ear is stimulated. And here you see those responses plotted. And so you can see the effect of the dramatic effect of the contralateral inhibition to turn off the response when the sounds are in the contralateral field. Okay, so now let's discuss the processing of ITDs. And we'll begin by comparing the precision of timing in the auditory nerve fibers as compared to the bushy cells. We recorded from bushy cells by electropenetrations in the trapezoid body by recording from the axons. So we made a surprising discovery soon after we began making these recordings from axons of the bushy cells from the trapezoid body. The bushy cells were more precise than their inputs from the auditory nerve, despite the intervening synapse. So this slide shows a comparison of typical recordings from an auditory nerve fiber and a bushy cell matched in CF, so approximately the same CF, in response to pure tone stimuli at CF. So the dot rasters left and associated period histograms on the right show responses to 200 repetitions of that CF tone. Clearly, the bushy cell shows a more precise phase locking, as easily evident in these improved alignment of the dot rasters and the more sharply peaked period histogram. So phase locking is typically measured by the so-called vector strength, which we call R here, which is given here. The values are given here. So vector strength varies from 0, where the histogram is flat, to 1 when all the spikes fall into one bin. This shows a sample of period histograms for CF tones of 5 different auditory nerve fibers, 5 different spherical bushy cells, and 5 different globular bushy cells, approximately matched in CF. So going from low CFs, 300 hertz, to high CFs, 7 or 10 kilohertz. So the vector strength values are denoted here, and the CFs are denoted here. So clearly, you can see that these low frequency spherical and globular bushy cells have period histograms that markedly differ from those of auditory nerve fibers. Now, at these low frequencies, you can't distinguish spherical from globular bushy cells because they're simply giving a phase lock response. So to distinguish them, we recorded intracellularly from their axons and injected them with HRP to trace their axonal projection into the superior olive. And spherical bushy cells were defined as those axons projecting to the MSO, while globular bushy cells had axons that had a calyceal ending in the MNTB. To compare the population of auditory nerve fibers and bushy cells, here we plot maximal vector strength as a function of CF. All auditory nerve fibers fall into the region defined by the two lines taken from the data of Johnson and confirmed by our own recordings from the auditory nerve. So no auditory nerve fiber ever had a maximal vector strength greater than 0.9. And so you can see that below 1 kilohertz or so, the majority of bushy cells have vector strengths that exceed that of auditory nerve fibers. So here we see an example of a physiological specialization in a circuit that is computing ITDs, and obviously this specialization can enhance the temporal acuity of the system. Note that for both the auditory nerve and bushy cells in the cat, phase locking essentially disappears above about 3 or 4 kilohertz. Now let's consider the responses in the MSO and LSO. The classical description by Goldberg and Brown of MSO cells is that they are high-resolution coincidence detectors. This slide shows evidence for this from recordings in the MSO to tones at CF, if for this cell conveniently 1,000 hertz, as a function of ITD. Our convention is that positive ITDs correspond to the contralateral ear leading, as shown by this schematic. Clearly, the cell is very sensitive and modulated strongly by ITD with a cyclic response that has a period corresponding to the stimulus frequency, that is 1,000 microseconds. So at each of the peaks in the response, the stimuli are in phase, just shifted by one complete cycle. This demonstrates that it is actually the interval phase, not the ITD, to which the cell is sensitive. While the cell responds best to binaural stimuli, it also shows a weaker response to monaural stimuli, shown by the arrows in down here, and plotted as period histograms on the right to contralateral ear alone and ipsilateral ear alone. These monaural responses are phase locked with a mean phase indicated by the downward arrow. And you can see, as expected from the anatomy, the ipsilateral input arrives slightly earlier, about 150 microseconds in this case. The coincidence model would predict that optimal response should occur if we give the contralateral ear a lead of that amount. And indeed, the peak of the ITD curve is approximately, but not exactly that amount. So this plot shows a test of the coincidence model for our population of MSO cells. On the ordinate here is the predicted phase from the monaural responses and the actual peak phase in the binaural responses. The model would predict that all points would fall on the 45-degree line, and you can see they're pretty close. Now, unfortunately, it has proven very difficult to record from the MSO. So most studies of binaural processing have been done in the central nucleus of the inferior colliculus, which is admittedly a poor surrogate for the MSO. Nonetheless, much progress has been made studying the IC. All cells in auditory nuclei respond over a range of frequencies. So it is of interest to look at the ITD curves for each cell over that range. So this slide shows a sample of 4 cells in the IC with each of the tone delay curves plotted on the same axis and normalized to the peak. In 1966, Rose, Gross, Geisler, and Hine proposed the concept of characteristic delay. which was defined as cells in which the periodic ITD curves for different frequencies reach the same relative amplitude. The CD would be the delay at which all of the ITD curves have a common peak, as shown by, appears to be the case in this cell, or a common trough in this case. For this cell, it's harder to say whether there's a common trough or perhaps a common peak or somewhere in between. And for this cell, even more difficult. So Shig, Kawada, and I indeed found it difficult to classify cells as satisfying characteristic delay by simply eyeballing responses, such as shown here. So we came up with a way to quantify whether cells exhibit characteristic delay by making use of the well-known property that a system in which there is a time delay, the phase versus frequency function will be linear with a slope equal to the delay. So the test of whether a cell satisfies CD is whether the interval phase versus frequency plot is linear. To visualize these responses, it is easiest to consider a model with unnatural triangular ITD curves, as drawn here. So there are three models here, one in which there's a common peak, or a common trough, or a common point in between. Cells that have a linear interval phase versus frequency relationship are defined as exhibiting CD, and the value of the CD is given by the slope of the phase frequency plot. Here you can see for the one that has a common peak, we have a slope that corresponds to the value of the common peak. Furthermore, the phase intercept, what we call the characteristic phase, is 0. So the CP tells you where the common point is. If the common point is at the trough, then the phase frequency plot is also linear, but now has a CP near 0.5 corresponding to one input out of phase. And again, the slope of the line corresponds to the point of the common trough. This cell has a common amplitude, but its characteristic phase now is not 0 or 0.5, but rather somewhere in between. And again, the slope corresponds to the point of that common amplitude. So if we go back now to the example shown on the previous slide, the interval phase versus frequency analysis are shown in red, with the point of CD plotted by the arrow. As expected, this cell, which appeared to have a common peak, has a CP near 0, and the common peak occurs at about 113 microseconds, corresponding to where one would eyeball it. This cell, which appeared to have a common trough, has a CP near 0.5, and the CD corresponding to approximately that point. This cell had a linear phase frequency plot, but its CP is not 0 or 0.5, but somewhere in between. And one would be hard pressed to identify this point as the common point. Finally, this last example had a nonlinear phase frequency plot, so it did not satisfy characteristic delay by this definition. Now, pure tones are rarely heard in nature. More natural but well-defined signal is broadband noise. So this slide shows responses of 9 inferior Cliquot cells to ITDs of broadband noise, shown by the solid line and the asterisk, plotted compared with the response predicted from its response to pure tones obtained by simply averaging all of the individual tonal curves. That's given by the dotted line. So you can see for these 9 cells in the IC, there is a surprisingly good correlation between these two measures. So the response to broadband noise can be predicted from a linear summation of its response to pure tones over the bandwidth of the noise. Now, if we do CD analysis on cells in the superior olive, So here's an example from the MSO. We can see that the cell in the MSO appears to have a common peak. If you look at these, we didn't normalize the peaks in this plot. And the CD analysis shows a CP near zero, a slope of 33 microseconds, which is very close to where the noise delay curve has its peak here, in this case, at 36 microseconds. Low frequency LSO cells are also ITD sensitive and show a characteristic delay with the CP now near 0.5 as expected. And you can see the slope of the line predicts a characteristic delay common trough at about 500 microseconds, which is close to what we found with the noise delay curve. Now, most models of binaural hearing incorporate cross-correlation to mimic the coincidence in the MSO. Here, we wanted to test this idea by studying responses of cells in the inferior colliculus to noise stimuli that differed in the degree of interaural correlation. So we generated noise stimuli by starting with two independent noises, call them A and B, delivering A to one ear and some combination of A plus B to the other with different ratios of A and B. We could thus generate noise stimuli with interaural correlations between one, which identical noises to the two ears, so A to both ears, or zero, A to one ear, B to the other, or any value in between. In addition, we could generate interaural correlations of -1 by delivering A to one ear and -A to the other. So you can see that this ICC cell responded with a systematic change in the degree of modulation as the interaural correlation changed from 1 to 0. And when the interaural correlation was -1, the response is essentially 180 degrees out of phase. So the plots on the right show for a sample of 10 cells in the IC, plots of degree of modulation as a function of interaural correlation. And you see a very systematic relationship for every cell. So this is exactly what you'd expect out of a machine that's computing cross-correlation. In addition, we examined the effects of delivering the same tokens of noise, but phase shifting the noise to one ear. By digital filtering, we phase shifted the noise to one ear in increments of 1/8 of a cycle. So here's the normal response, identical noises to 2 ears. Now phase shifting the noise to the ipsilateral ear by 1/8 of a cycle, 1/4 of a cycle, 3/8 or half a cycle, or phase shifting by 1/8 to the contralateral ear, 1/4 of a cycle, 3/8. And similar manipulations are shown down here. You can see the remarkable machine-like properties of the two ICC cells under the 8 different conditions. Basically, all ITD sensitive cells we tested in the IC behaved in a similar way. So the nervous system appears to achieve this high resolution ITD sensitivity by employing a process that is very similar to the mathematical process of cross-correlation. Interestingly, naval engineers working on a similar problem to localize submarines in open ocean have developed methods which also involve cross-correlation from wide underwater microphone arrays. So the brain seems to use a technique which is similar to what humans have developed, human engineers have developed. Okay, now let's move on to ILD sensitivity and the LSO. Well, we know that classically LSO cells are known to be sensitive to ILDs, but I've shown you that they are also sensitive to ITDs. I showed you an example of a low frequency LSO cell that's sensitive to ITDs, a pure tones. I didn't have time to show you evidence that high frequency LSO cells are also sensitive to ITDs, providing you give them a time varying envelope, for example, like a AM stimuli. So when stimuli are delivered in free field, what is the relative effectiveness of each of these three acoustic cues, ITDs, ILDs, and spectra? So we made use of virtual acoustic stimuli to manipulate each of the three cues in isolation to study that question. So for each point in space, there is an unique ITD, ILD, and spectra combination. With digital manipulation, we can set one of those cues to be constant while the other two vary, or we can vary that one cue while the other two cues are held constant. So this shows you responses from a single LSO cell, and each panel shows a results of manipulation of one of those cues, ITDs, spectra, or ILDs. So for example, this panel here, manipulating ITDs, we see first the normal response, filled circles, shows the expected response in the ipsilateral sound field. And that response is plotted in each of the three panels. The red delta ITD response is obtained by varying ITDs while holding the other two cues constant to their values at 0 degrees. And you see that response is unmodulated. While the open circles show responses when ITDs are held constant and the other two cues are varying. That response is nearly equal to that of the normal, right? So both of these results suggest that ITD is not a salient cue for the cell. Similarly, the responses when we manipulate spectra in analogous ways, you get the same kind of similar responses, right? On the other hand, when only ILDs are varied, the red curve, you can see a response which is very similar to that of the normal. And when ILDs are held constant, the cell is unmodulated. Right? So these results suggest that ILDs are really the salient cue, right? And generally speaking, the population of LSO cells we studied behave similarly. So the conclusion of this experiment was the rather boring one, that ILDs are the most salient cue for LSO cells, which fits the classical view. So if that's true, then why is there the largest synapse in your brain in this circuit? So the calyx of Held has become a model system for studying the biophysics of synapses. Yet we don't understand why such a highly specialized structure has evolved in a circuit that appears not to be dependent on temporal effects. So the answer to this might be found by looking at responses of the LSO to ITDs of transient stimuli, such as clicks. So ITD functions to clicks in the LSO have a plateau with a narrow window of inhibition when the contralateral inhibition is timed just right to inhibit the ipsilateral excitation, as shown by the schematic up here. On either side of this window, the excitation arrives either too early or too late to be inhibited. So note the steep, very steep ITD sensitivity of this LSO cell along both sides of this temporal window. This cell also illustrates one of the remarkable features of the IE interaction in the LSO, namely the contralateral inhibition arrives nearly synchronously with the IPSI excitation, even though it has to travel farther and has an additional synapse. So Boudreau and Tucitani in their seminal studies of the LSO had already made this observation, since repeated by many others. So the calyx of Held may serve to minimize the delay of the contralateral inhibition to the LSO and make it exquisitely sensitive to ITDs of transients. Okay, I will finish by describing some of our behavioral studies of sound localization. We trained cats to look at visual and auditory targets. The cat faced an array of speakers. Each speaker had an LED at the center, of course, so corresponding visual or acoustic targets could be presented. Eye position was monitored by suturing a small coil of wire on the eyeball and placing the cat in an alternating magnetic field set up by coils embedded in this cube. And we simply use techniques that are developed by oculomotor physiologists. The cats worked for a food reward under operant conditioning, and eye position was continuously monitored under computer control. And the reward was delivered if the eye position was within an electronic window centered on the target. So typical trials began with an LED straight ahead turned on. Cat looks at the LED, and after some delay, unpredictable delay, the target is turned on and the cat orients to it. So we initially did the experiments with the head fixed, but we soon found that localization performance was much better when the head was free and we measured gaze or eye position in space. Right? So this shows the eye movements to visual stimuli on the left, auditory ones on the right. The fixation LED is on until time 0 when the target is turned on. The location of the targets are given by the little arrows on the right. Visual trials are shown on the left, auditory on the right, and we plot here now the vertical gaze shifts to stimuli varying in elevation on the vertical axis, or horizontal gaze shifts to targets varying in azimuth. You can see that the saccades to both visual and auditory targets are both very accurate and precise. Right? They hit the target, and there's relatively little scatter. And these are quantified in the next slide. So here are scatter plots of localization of short and long duration visual targets, short and long duration auditory targets. And we plot the vertical gaze shifts to the targets varying in elevation, horizontal gaze shifts to the targets varying in azimuth. And the targets are color coded. So the gain or regression line of the responses shown by the red line is a measure of the accuracy of the gaze shift. And that's given by the slope of the regression line. Perfect performance would be a slope of 1. The delta value measures the precision or the distribution of responses about the mean and is computed as the standard deviation of the residuals. So in general, performance is better for the long duration targets than the short for both visual and auditory cases. And it's slightly better for visual stimuli than it is for auditory, although that difference is relatively minor. So this slide compares the performance of one of our cats to some other sound localization measures in other studies in the barn owl and 2 studies in human. So it looks like our cats are able to localize just as well, or maybe even better than these other subjects. All of the responses are plotted on the same scale here. Now, to test the effects of spectral cues, we studied localization of narrowband noises, 1/6 octave wide, centered on 6, 8, 12, and 16 kilohertz in the region of the first notch. So these narrowband stimuli will have impoverished spectral cues while maintaining ITDs and ILDs. And here we plotted the scatter plots for vertical and horizontal targets separately, just for ease of looking at them. The azimuthal localization is degraded. You can see that these slopes are not quite as good as it is for broadband noise. But they're pretty much near normal. but vertical performance is significantly worse. Right? The low gain values for vertical localization. In fact, the cats look like they're essentially guessing in some of these cases. So this supports the idea that spectral cues are crucial for localization and elevation. So let me conclude with some observations about what the cat is doing with its pinna while localizing sound. So first, we examine pinna movements when the head is held stationary. So here's a short video taken while the cat is working on the visual localization task, so you can see the target. We pasted some white markers on the edges of the pinnas just to help you see the movement. And all the trials begin by turning on the LED straight ahead. And you can see the cat orients to it and then moves in a consistent way depending on the location of the target, which is indicated afterward. So watch the right ear and try to predict which direction the target was. Noteworthy is that the cat is being rewarded for its eye position, which you can't see, but not for pinna position. It can do whatever it wants with the pinna and still get a reward. But you can see that you can predict the position of the target simply by watching the pinna, which orients to all the targets except those on the contralateral side. So to quantify these pinna movements, we surgically implanted a coil at the back of the pinna, so that in addition to the eye coil, we had a pinna coil. Here are plotted the movements of the eye on the top and the pinna, the vertical component, and the horizontal component of the pinna movement on the bottom. In these trials, the target was turned on before the LED went off. The cat was trained to make the saccade after the LED was turned off. And you can see that, again, the eye position is quite accurate to the target, and the pinna moves consistently to rightward for this rightward target, both visual and auditory stimuli. One difference is that the pinna moves for the visual stimuli about the same time that the eye moves, but for the auditory stimuli, the pinna moves right away and with a second movement when the eye moves. So an important point is that, as we saw in the movie, the pinna responses are consistent and reproducible, even though cat was not being rewarded for moving the pinna. Now, what happens when the head can move? Since the pinna rides on the head, it should turn when the head turns, unless there is some compensation. Now we have three coils, one on the eye to measure gaze, that's the green trace, one on the left ear, blue trace, and one on the head, the black trace. And in addition, we calculate ear on head by subtracting the head from the ear position. You can see that the ear moves first, right, and maintains its position towards the target, even though the head, which is moving slower with a slightly longer latency, even though the head is moving. So the only way it can maintain its position in the face of the head movement is by counter-rotating the ear on the head. If we take the peak ear on head velocity and divide by the peak head velocity, we can calculate a gain of we call this the vestibulo-auricular reflex. This gain, in this case, is nearly perfect compensation of -1. So we call this the VAR because it shares many kinematic characteristics of the well-known VOR, vestibulo-ocular reflex, in which the eye counter-rotates to compensate for the head movement to keep the retina stabilized in space. So the ear must counter-rotate on the head to maintain that constant position. So the VAR differs from the VOR in that it is only active for head movements towards the ipsilateral side. And it's simply easiest here just to look at the red traces. This trace is what I showed you on the previous slide. So these are 6 targets varying in azimuth. You can see that we're measuring the left ear, and for all the 3 targets on the left side, we see this compensatory VAR. But when the targets are on the right side, the ear simply rides with the head, and there's no VAR. So not shown are some control experiments in which we passively rotated the head and saw that the VAR was still active, thereby suggesting it is vestibular in origin and not due to an efferents copy. So it's not every day that one can discover a new reflex. So I'd like to conclude by acknowledging the many wonderful colleagues, students, postdocs, faculty on leave that I've worked with over the past 40 odd years. They are really the ones responsible for this work. I've also been blessed by working with the faculty and staff of the Department of Neurophysiology at the University of Wisconsin, as it was named when I first joined, who educated me on all things auditory. Also, I thank NIH and NIDCD for funding the research over these many years. Thank you.